Neurons exchange information through electrical signals. These are generated at the beginning of the axon and travel down towards the site of connection with other neurons. Scientists had a hard time studying the electrical signaling happening in the neurons until this funky animal was found. This squid has a giant axon, which made it easy for two scientists, Hodgkin and Huxley, to finally measure and describe what was going on in the axons of neurons. Across the neuronal membrane, charged ions, potassium, sodium, chloride, and calcium, are present at different concentrations outside and inside the cell. In the membrane, ion channels are present and let specific ions flow through them. This way, ions tend to flow down their concentration gradient because they want to equilibrate their inside and outside concentrations. But because different channels have different specificity and permeability to distinct ions, charges are never evenly distributed, and this leads to a difference of potential across the membrane. This is called the resting membrane potential, which is around -70 mV. We say that the membrane is polarized. If this potential goes above a certain threshold, then an action potential is fired. According to Hodgkin and Huxley, firing begins with the opening of sodium channels. This produces a massive influx of sodium ions. The rapid accumulation of positive charges increases the membrane potential. This is called depolarization. The depolarization lasts for a short time until the sodium channels close and inactivate. During this period, it is impossible for sodium channels to reopen. Following this, potassium channels open and potassium ions flow out of the cell, reducing the positive charges within the cell, repolarizing the membrane even past its resting potential value. This last phase of the action potential is called after hyperpolarization. With the closing of potassium channels, the membrane then goes back to its resting state. So that's a pretty clear and simple model. And because of it, it was also once believed that action potentials were all the same. But now we know that, in mammals, action potentials can be quite different from one another, and their generation process is much more complicated than what Hodgkin and Huxley described. So what makes action potentials so different? A crucial factor is the variety of ion channels and their corresponding currents. Let's start by discussing the sodium channels. Many different types of sodium channels are present throughout the body. A particularly interesting sodium channel was discovered in the cerebellum. This type of channel does not inactivate in the same way as other sodium channels. Researchers have discovered that a unique blocking particle plugs up the ion channels as the cell depolarizes, preventing the normal inactivation gate from closing. When the cell repolarizes, the blocking particle leaves and sodium ions flow once again. This flow is known as resurgent current. This mechanism makes channel inactivation shorter, and the flow of sodium slightly depolarizes the membrane, so the time before another action potential can occur is also shorter. This is important for fast spiking neurons that fire action potentials at a high rate. Scientists have found that these particular channels are involved in chronic pain, arrhythmia, and muscle disorders. Calcium channels are cool too. They can directly or indirectly shape action potentials in mammalian neurons. There are several types of voltage-gated calcium channels, which can be activated by high, intermediate, or low depolarization voltages depending on their types. Activation of calcium channels leads to a large inward calcium current that would make action potentials broader. But surprisingly, in some neurons, calcium channels result in narrower action potentials. This is due to colocalization of calcium channels with potassium channels that can be activated by calcium ions. The consequent potassium current generates an outward ionic flow, overriding the inward calcium current effect. The net outcome is a narrower action potential. Potassium channels are even cooler, because they're extremely diverse. A single type of potassium channel can mediate both the firing pattern and the shape of the action potential. For example, the Kv3 channels, activating during the repolarization period. At the end of the depolarization phase, Kv3 channels open very fast, which leads to relatively narrow action potentials. The deactivation of Kv3 channels is also extremely fast, producing very short after hyperpolarization. At the same time, these channels have high activation thresholds. Thanks to all those characteristics, the neurons bearing those channels are better at high-frequency firing. KV3 currents don't work in isolation. They also reduce inactivation of sodium channels, making the recovery of sodium currents larger and faster, therefore mediating the resurgent sodium current that we talked about earlier. KV3 currents also interact with other ion channels, regulating sodium and calcium signaling involved in burst firing behavior. Disturbances in KV3 function can cause various motor dysfunctions, including ataxia in humans. So, we have established that different ion channels can modulate the shape and the firing patterns of action potentials. But the amount of neurotransmitter released by the action potential can also be modulated. Interestingly, this can be done by currents that are too weak to trigger an action potential by themselves, the so-called subthreshold currents. As electricity travels down a wire, these currents travel down the axon, without having to be continuously boosted, like it is the case for the action potential. Once they reach the presynaptic terminal, they impact the message of the action potential by facilitating release of neurotransmitters. But hey, it gets even more complicated, because this story is actually not just about neurons. I swear, for a long time people believed that neurons were the only cells responsible for the electrical signals sent to other neurons in the brain. It's only more recently that we started to understand that glial cells, the proverbial glue of the brain, have more of an impact than what was thought. We now know that by releasing glutamate, the glial cells that line an axon can broaden action potentials and electrical signals as they travel down this axon. We used to think the action potentials were all the same, created from just a handful of ion channels, on or off, reproducible, consistent. It turns out the action potential is not so simple. From the multitude of ion channels to the small currents traveling passively down the axon and the influence of glial cells, a variety of complex factors shape the message that is sent to the next neuron. Perhaps it is time to admit that few things in life are black or white, all or nothing.